Okay. So I'm going to start with the calculations. Of the bacterial population. Sounds good?
Yeah.
I'm going to give one example of each, so we don't spend too much time on this. Okay. And let's say, so here's what you're given. Okay. You have 10 cells at the start. Okay. Generation time is 30 minutes. Okay. Growth time, well, let's make it 3 hours. Okay. Find the number of cells. Are we good so far? Well, I presume we are. Okay. So what we start with, we say that the generation and the number of cells. So the generation 0, number of cells is 10. Okay. Remember that every generation, the number of cells doubles. Okay. So there are several ways to skin the cat. I'm going to give you the simplest one. So generation 1, you're going to have 20 cells. Okay. Time is 30 minutes. Generation 2, you have 40 cells. That time is 30 minutes again, so it gives you 1 hour. Okay. Generation 3, the number of cells is 80. That's another 30 minutes. Generation 4, the number of cells is 160, which gives you, and another 30 minutes is spent, which is 2 hours. Okay. So first hour, second hour, right? At the 5th generation, the number of cells is 320. Another 30 minutes. The little apostrophe, it shows the minutes, okay? And then at the 6th generation, you have 640 cells. And another 30 minutes give you third hour. If you look at the conditions, 3 hours, right? So we completed our 3 hours. How many cells do we have at the third hour? 640. That's your answer. Now, I suggest you unmute your microphones because I want to know if it makes sense to you.
Yeah, it makes sense.
Okay. Now, give me 1 second. And I will continue talking about generation time.
Okay.
So for generation time, you're essentially using a very similar approach. Okay. Does that make sense?
What did you say?
You used a very similar approach.
Mm-hmm.
You just consider it in a slightly different way. So we're going to call it calculations. Of I'm sorry, I have a brain fart right now. Calculations of the generation time. Okay, so how do you approach that? So what you need to do, let's say you have some given, okay? What are you given? You start with 100 cells, okay? You end up with 12,800 cells. So far, so good. And it takes 2 hours, 20 minutes to get there. Find the generation time. So step 1, find the number of generations. Okay? Make sense so far? So how do you do that? There are 2 ways, okay? So I'm going to say fast way. Step A, divide final number by the initial number first. So you divide 12,800 by 100, which gives you 128. Okay? Then take a logarithm with the base of 2 from 128. And don't worry, I'm going to tell you, you can do it on your calculator. If you feel shaky and uncomfortable about logarithms, then you have the slower, not the slow, but the slower way. Okay? Divide sequentially 12,800 by 2. Let me show you what I mean. And we divide 12,800 by 2, and we get 6,400. Okay? So this is the step 1. Does that make sense? Folks, I would really appreciate some feedback. Huh?
On the fast way,
No.
where you said after we divide 12,800 by 100 and we get 128, and then you said take the logarithm with
With
base
a
of
base of
2.
2. Now, if you feel uncomfortable around logarithms, you can forget the fast way.
Is that a button or something that's to be on the calculator, or?
It's okay. Here's the deal. You're probably going to be easier. It's probably going to be easier to just take a slower way. Okay?
Okay. Mm-hmm.
Look at this. So take a first step. You divide 12,800 by 2, you get 64,000.
Mm-hmm.
Good?
Yeah.
Then you take 64,000 divided by 2, you get 3,200. That's a step 2. Am I clear?
Okay.
You get 3,200 divided by 2, you get 1,600. This is the step 3. You get 1,600 and divided by 2, you get 800. This is the step 4. You get 800 divided by 2, you get 400. This is the step 5. You divide 400 by 2 and get 200. This is the step 6. You get 200 and divided by 2, that gives you 100. This is the step 7, right? So basically, you go by division, you go backwards in terms of the generations, okay? Does that make sense?
Right. Until we get back to the start cell number.
So this
Mm-hmm.
is the number of generations.
Right.
Do you follow me?
Mm-hmm.
Yes, I do.
Now, total time is 2 hours, 20 minutes, or 140 minutes. You can calculate it yourself, okay? So your
Mm-hmm.
generation time will be total time divided by the number of generations. Am I clear?
Yeah.
Yes.
Which means 140 minutes divided by 7, which gives you 20 minutes. So this is your generation time. Does that make sense?
Uh-huh.
Okay, we're good?
Yeah.
So what I would do if I were you, I would take, well, you don't really have to put it on the paper, but you can essentially just lay out the exponents of 2 on the paper. Okay,
Mm-hmm.
I would also totally do this. Um, Draw. Oh, that's terrible, isn't it? Okay. No, I'm going to kill it. So what I want to show you is how to approach some of the things and make a use of make use of the what I wanted to say, sorry, of the scratch paper. Okay, does that make sense?
Mm-hmm.
Mm-hmm.
Okay. So this is what I would do if I were you when we're talking about the growth curve. So I would just draw the growth curve for its own benefit, okay? And then I would name individual steps. One is the log phase. Not much is happening, right? 2 is the log phase. Um, dying is much less than dividing, right? Waste is much less than nutrients. 3 is the stationary. Dying is roughly equal to dividing, and waste is less, not much less, less or equal to nutrients. The 4 is the death phase. So number of dying cells is much more than the number of dividing, and waste prevails. Okay, we're good so far, folks?
Yes.
Yeah.
Okay. Then and then you can answer a whole bunch of questions here. Then we are moving on to the conversation of cell counting. Okay. And remember, you can basically just classify them based on the live cells, right? And you have, just as a reminder, right? You have direct cell count with a Petrov house or chamber. So please make sure that you know kind of what is happening there. Okay. You have
What is happening with direct cell count?
during the direct cell count, yes. So
Okay.
like you doing it using Petrov house or chamber in the microscope.
Okay.
Then you have Bible cell count, and you have most probable number method. And remember that in most probable number, when you have a number, it's not an actual number of the cells, it's the probability that is the highest. Like if the number is like, if your MPN, most probable number, is 80 with a certain confidence limits or confidence interval, then the probability is the highest for 80. Okay, does that make sense?
Right. Yeah.
Then all cells will be Folder counter and turbidimetry. Okay. And remember, cold counter, what happens? Turbidimetry is not even the cell count. So we're measuring optical density or light absorption. Okay, so that brings us to the concept of, so let's talk about, like, communities. Okay. And so my goal for uh, this week is to review the conditions for growth and talk about some of the hold on. I'm just looking who's here. Okay, you two of you. Okay, it's fine. So communities, microbial communities. So what you got to know is that we have obligatory relationships, all right? So And what I'm talking about is mutualism, commensalism, and parasitism. So let me explain what you need to know. You have to understand the relationships by definition, like literally by definition. Am I clear? This is what I'm going to ask you. Does that make sense, folks?
Yes.
Yes.
Okay. So parasitism. In terms of parasite, be able to distinguish between endo and ectoparasites. So like helminths are endoparasites and lice are ectoparasites or ticks or fleas.
Should we know examples?
Well, you have to be able to appreciate it. I don't want you to know the species name, but if a tick or a lice is an example, then yeah, you have to know them. Like you have to process the information. You see what I'm saying?
Mm-hmm.
Like if I ask you, which of the following is ectoparasite, and I give you like a virus, a bacteria, a protozoa, or a lice, you tell me
Mm-hmm.
all lice, right? And then non-obligatory relationships. So that will include synergism and antagonism. Okay, so again, know the definitions. Now I'm going to throw biofilms in here. As I mentioned before, I want to reinforce it. Reinforce it. Know the process. Okay, like primary colonizers, extracellular polymeric substances, secondary colonizers, um, when they reach critical density, they start producing quorum sensing molecules, which enable a communication between the cells in a biofilm. Okay. Question so far.
And you said understand the process of biofilm.
This is as far as it goes.
Okay.
Um, what I'm saying is, if you just memorize this schematic, it may be hard for you to answer the questions. But if you understand what's going on, I'm not asking you what are the types of quorum sensing molecules. I'm not asking you, what are extracellular polymeric substances. But like, for example, what is the role of extracellular polymeric substances? Okay, for instance, they protect against desiccation and predation. Does that make sense?
Yeah.
So that's what I'm asking.
Okay.
I remember I don't if I can, I don't ask about memorization.
Mm-hmm.
Okay. Anyway, let's move on because I want to cover some more ground here. Um, so nutrients. Know the concept of growth factor. Essential nutrient. So know the definition. Know the following. So carbohydrates, carbon, hydrogen, oxygen. Okay. Proteins, carbon, hydrogen, oxygen, nitrogen, sulfur. DNA, RNA, carbon, hydrogen, oxygen, nitrogen, phosphorus. Lipids, carbon, hydrogen, oxygen. Phospholipids, carbon, hydrogen, oxygen, phosphorus, obviously, nitrogen in many cases. Okay? So far, does that make sense?
Yeah.
Yes.
Okay. Biogeochemical cycles. I'm gonna, well,
That's the
okay.
carbon cycle and those cycles.
Yeah, so terms that you need to know. So, okay, I'm gonna put it this way because I don't want to repeat what I said before. Terms to know. Okay, so let's start with the carbon cycle. How about that? Okay, so carbon cycle. Cellular respiration, carbon oxidation, photosynthesis, carbon fixation, methanogens, and methanotropes. When I say know the terms, understand what they do. Okay, like what happens during cellular respiration? Organic carbon is oxidized to inorganic. What happens during photosynthesis? Inorganic carbon is reduced, fixed into organic. What do methanogens do? They generate methane. What methanotropes do? They use methane as the source of carbon. Okay, does that make sense to you? So you can totally expect those questions, just basically the terminology. Carbon cycle. Okay, next one I want to chat about is nitrogen cycle. This is very straightforward. Uh, Molecular nitrogen to ammonia. Ammonification. Okay,
Do we have to know like the symbols for it, or are we gonna know it just like this?
I can't hear you.
I was saying, do we have to know the symbols for like NH3 and NO3
If you
minus?
come to me and ask me what does NH3 mean, I will immediately tell you.
Okay.
So whether you memorize it as the formula or the words, I will give you the information that is otherwise. Does that make sense?
Yes.
So if you see N2, you can come to me and ask, what is that? And I will tell you, this is the molecular nitrogen. Okay?
Mm-hmm.
Ammonia 2 nitrate, nitrification. nitrate to molecular nitrogen, denitrification. Okay, does that make sense? Finally, for the sulfur cycle, okay, sulfate to hydrogen sulfide, anaerobic respiration. Hydrogen sulfide to sulfur, or sulfate, anoxygenic photosynthesis. Okay, does that make sense so far, folks?
Yeah.
Okay, so what I want to cover, a couple more things. So I want to cover the some requirements for growth. Okay. And give us some idea what to focus on. It's all definitions. So let's start with this. Temperature. You have 5 groups. Psychrophiles grow below 4 degrees Celsius only. Okay. Psychotrophs grow at room temperature, can slowly grow in a fridge. Does that make sense?
Mhm.
Mesophiles grow at body temperature. Now, will I ask you about the specific temperatures, like numbers? No. But you have to know, like if I ask you sacrophiles, where would they grow? You tell me they would grow in the fridge only, not at the room temperature. Does that make sense?
Yes.
Thermophiles grow at scalding water temperature. So like water is hot, but not boiling, okay? Give it a second. It's the system problem. Extreme thermophiles grow at boiling water temperature. Does that make sense to you all folks?
Yeah.
pH requirements. This is very easy. Neutrophils, which are most human pathogens. pH between 6 and 8. Acidophiles, pH lower than 6. Okay, does that make sense so far? And alkaliphiles, pH more than 8. Now, some of the stuff like, miscellaneous. Zerophiles, dry environment. You with me?
Yeah.
Aerophiles, high pressure. Osmophiles, high solute concentration, any solute. Now, within barophiles, within osmophiles, there are halophiles, high salt, sodium chloride concentration. Okay, we're good so far? Well, I presume that we're good. And finally,
Can you hear me?
yes,
Oh, okay.
oxygen requirements. This is probably the more complicated part. So, what can you expect here? You can expect questions like, human pathogens. What is their pH requirements? And I think, okay, humans, neutrophiles, right? Or which microorganisms grow at the temperature of the boiling water? And tell me, extreme thermophiles, right?
Mm-hmm.
You with me, right? Okay,
Yep.
fantastic. So, oxygen requirements. So, 5 categories. I'm going to put them here: obligate, aerobe, obligate, anaerobe, uh, air tolerant, anaerobe, facultative anaerobe, and microaerophile, which is also called the Goldilocks. Okay, so obligate aerobe. If you look at the tube growth, okay, the growth is on top. Does that make
Mm-hmm.
sense?
Yep.
Needs oxygen.
To
The
grow.
metabolism is aerobic respiration. And in terms of the enzymes that neutralize reactive oxygen species, superoxide dismutase, peroxidase, catalase. Okay, obligate anaerobe, the very bottom.
Mm-hmm.
Oxygen is toxic. Metabolism is usually fermentation or, well, let's stick to fermentation actually. Enzymes, usually none of the three. Okay,
Mm-hmm.
air tolerant anaerobe, growth all over. Okay,
Mm-hmm.
oxygen is okay, not required. Okay, metabolism, fermentation, or anaerobic respiration. Usually superoxide dismutase. Okay.
Mm-hmm.
Facultative anaerobe. More growth on top, but also growth throughout.
But it grows more at the top.
It may use oxygen, but may switch to fermentation. Not may, but can. Okay. Can use oxygen, but can ferment as well. Okay.
Mm-hmm.
Usually aerobic respiration plus fermentation. Usually superoxide dismutase and catalase. Okay, and microaerophiles but require precise small oxygen concentration. Okay,
Mm-hmm.
metabolism is aerobic respiration. Usually SOD, superoxide dismutase, plus catalase or peroxidase. Okay, we're good so far?
Yeah, I just got one question.
Go ahead.
For obligate anaerobe, there's no enzyme.
Enzymes that, no. So usually none.
Okay.
Enzymes that neutralize reactive oxygen species. Superoxide dismutase neutralizes superoxide anion. Catalase, and peroxidase neutralize hydrogen peroxide. That's all you need to know, really. I'm not asking about the reactions. Okay? Do you have any
Mm-hmm.
questions?
Nope.
This is as much as I wanted to cover today.
Okay.
If you have no questions, so this will be posted sometime tonight. Okay?
Mm-hmm.
And yeah, that's about it.
All right, we doing this again Monday?
Yeah,